Hello, everyone from Regensburg. I'm Neva, the head of the Biochemistry tree at the University of Regensburg and a former group leader and current affiliate professor at the Helmholtz Institute in Würzburg. It's my pleasure to moderate this RNA collaborative seminar. Today we have a joint seminar together with the Shanghai RNA Club. I will be moderating the first talk by junior professor Jens He. Jens completed his PhD in Würzburg and after a postdoc at the Weizmann Institute in Israel, joined HIRI as a group leader, and his talk will be about insights into the role of RNA in bacterial immunity and phage replication. So take it away, Jens.
Okay, so
sharing your slides, let me quickly announce that please write your questions in the question and answer and don't wait until the end of the seminar. We'll be reading the questions aloud, and if time allows, then we'll be also contacted otherwise by Jens. So thank you. Take it away. Thank you, Jens.
Yeah, so thank you, Neva, for the introduction, and also welcome everyone in the seminar. I'm Jens, and I recently started my own group at the HIRI, as Neva just mentioned, so today's talk will be a bit preliminary. But I want to start by just giving you this introductory slide showing that, as you all know, basically every organism on Earth can be infected by viruses, whether that's us humans or plants, but it's also bacteria that can be infected by viruses, which are called phages, as you can see here. And these bacterial viruses are actually extremely diverse. So if we look at different families of phages, we can see that they can have filamentous phenotypes, they can be icosahedral, they can have tails, they can have membranes, and so on. Also, the genomic material that they have is very diverse. So we have single-stranded or double-stranded DNA as a genomic material, as well as single-stranded or double-stranded RNA. So with this huge diversity of viruses that bacteria have to deal with, there's a simple question that can be asked, and this is, how do bacteria actually protect themselves against phage infection in order to not just succumb to the viruses that constantly infect them? And one of the ways bacteria do this is by using so-called defense systems to actively defend against phage infection, similar to what eukaryotes have in their immune systems. And one of the most well-known and most widespread of these defense systems is restriction modification systems that you probably all know from cloning experiments in the lab. And these restriction modification systems, they can recognize and cleave foreign invading DNA, for example, from a phage genome. And by cleaving this genome that is being injected by the phage, the restriction modification enzyme can stop the infection. A similarly well-known defense system that bacteria use is CRISPR-Cas that also recognizes invading nucleic acids, recognizes them as foreign, and then is able to cleave them and thereby stop the infection. And obviously, similar to restriction modification systems, CRISPR-Cas systems have also been repurposed as really powerful molecular tools. But apart from these two types of defense systems that I just mentioned, over the last around 7 years or so, a huge additional variety of bacterial defense systems has been discovered. And many of them have actually been shown to be the evolutionary origins of human innate immune systems, like, for example, Gasdermin proteins or viperines or others. And nowadays, we know more than 250 different defense systems that bacteria use to defend themselves against phage infection. And I just want to highlight one such system that I was working on during my postdoc, which is the Bacterial ISG15-like defense system. So this is a 4-gene operon that contains a ubiquitin-like protein, an E2 protein, a D-ubiquitinase, and an E1 protein. And as you can see from these proteins that are encoded in this 4-gene operon, This system actually looks very similar to a eukaryotic ubiquitination system, and that was very interesting to us. So this is why we studied it in a bit more detail. So here I'm showing you just a simple plug assay. So basically we grow bacteria in a lawn and then apply phages in a zero dilution on top of this lawn of bacteria. And every dot you see here is basically a phage that causes lysis in this lawn of bacteria. And when we compare the control strain that does not express this defense system versus a strain that expresses the defense system, we can see that there's a reduction in the number of plugs that this phage is able to form. And when we look at more of these more different phages, we can see that some of them are actually almost fully inhibited by the expression of this Bill system in this bacterial lawn. So what was interesting about the system when we discovered it is that when we looked at the predicted structure of this ubiquitin-like protein in the system, we found that it contains two fused ubiquitin-like domains. And when we performed structural homology searches for this structure that we predicted, we found that it's very similar in structure to human ISG15, which is a ubiquitin-like protein, and ISG stands for interference-stimulated gene. And as the name already suggests, this ubiquitin-like protein in humans is actually part of the innate immune system. And the ISG15 protein gets covalently conjugated to many different substrates upon viral infection, and thereby is able to inhibit infection by viruses such as HIV and others. And this was very interesting to us that we found a bacterial defense system that is analogous to a human innate immune system. So we're wondering how this system actually protects against phage infection. And to make a long story short, what we discovered is that when we isolate phages that emerge from an infection experiment in bacteria that express this defense system, and we isolate them using cesium chloride gradients, we saw that there are two different populations of phages that arise from this system, which is not the case when we just do the same experiment on a control or a mutated system. And through an array of different experiments, we could show that what happens is that the bacterial defense system covalently attaches its ubiquitin-like protein to the very tip of the phage tail, as you can see here in these electron microscopy immunogold labeling images. And what this does is that it obstructs the receptor binding domain that the phage uses and which is located at the very tip of the phage tail to attach to the receptor of the host bacterium in order to infect it. And this is one of the ways the system actually protects against phage infection. And as I've mentioned in the previous slide, there's also a second population of phages that emerges from bacteria that express this defense system. And when we looked at those phages under the electron microscope, we saw that these phages were actually missing their tails. And similar to what I showed before, of course, the phage tail contains the receptor binding domain, meaning that also these phages are not able to infect the surrounding bacteria, and thereby the infection is blocked. So basically what happens with this Bill system is that if bacteria express it and they get infected by a phage, the phage expresses this tail fiber protein that is essential for the structure of the virion. And the bacterial ISG15-like defense system covalently conjugates a ubiquitin-like protein to this structural protein of the phage. And this leads to the generation of either tailless phages or modified phages that are unable to bind the receptor. After the host is lysed by the phages, these two populations of phages are released, but they are not able to infect the surrounding bacteria, and thereby the population survives. So this was a very interesting discovery that we made, but now I want to quickly switch gears and talk a little bit more about other defense systems, and in particular about bacterial defense systems that use non-coding RNAs as part of the defense mechanism. So as I've mentioned, over the last 6-7 years or so, more than 250 different bacterial defense systems have been discovered. And actually, several of them use non-coding RNAs to defend against phage infection. So one of the most well-known, of course, is CRISPR-Cas that uses a non-coding CRISPR RNA to guide the Cas proteins to the invading nucleic acid, and then allows it to specifically cleave the foreign DNA to block the infection. Other classic systems that contain non-coding RNAs are toxin antitoxin systems that use non-coding RNAs in order to either block the production of the toxin or inhibit the toxin directly, and thereby shut off the toxic function of the toxin. And then upon phage infection, the toxin gets released or produced, and this leads to abortive infection, thereby protection against the infection. Now there's several different additional systems that use non-coding RNAs for defense, and one of them are Retrons that I want to talk about in a bit more detail now. So Retrons were discovered already more than 30 years ago in 1989. And what's very curious about these Retron systems is that they produce a unique molecule called the msDNA, which is a hybrid molecule of RNA and DNA, which is covalently linked. And as you can see in this original gel image here, if you run this msDNA on a gel and then add RNAs, you still have a band indicating that this is the DNA part of the msDNA and this is the full hybrid molecule. So how does the cell actually produce this msDNA? So the Retron consists of two different genes, a non-coding RNA and a Retron type reverse transcriptase. And these two parts of the system of the Retron get produced. So we have a non-coding RNA and the reverse transcriptase. And the non-coding RNA folds into this very specific fold that base pairs the 5' end to the 3' end, which then brings together two guanosine residues that are branching and are opposing each other. The reverse transcriptase then can bind this stem structure here that is formed by the non-coding RNA and starts reverse transcription from the 2' end of this branching guanosine up here and reverse transcribes the MSD part of the non-coding RNA. At the same time, the RNA template strand gets degraded by the host RNase H, and in the end we end up with the mature msDNA molecule where, as I've mentioned earlier, you have a 2'/5' linkage between this branching guanosine and the cDNA, and then the 3' ends of the RNA and the cDNA base pair. In addition to these two genes, the non-coding RNA and the reverse transcriptase, Retrons always come with an accessory gene, or sometimes also more than one accessory genes, meaning that Retrons are actually tripartite systems. So for example, if we look at this type 1b1 here, we have the non-coding RNA, the reverse transcriptase, and then we also have an ATPase top rim domain fusion protein that is encoded as an accessory gene downstream of the reverse transcriptase in this case. And there's actually a huge variety of different Retrons that have all kinds of different types of accessory domains. And as I've mentioned, Retrons have been discovered more than 30 years ago, but actually until a few years ago, no one knew what their biological function was. And this changed when people, or more specifically the Sorek lab at the Weizmann Institute, figured out that Retrons actually function in anti-phage defense. So here you can see a plug assay again, as I've showed you for a different system before. And when you drop phages on the bacteria without a system, you can see clear plug formation. But then if you have bacteria that express a Retron and you do the same experiment, you see that there's almost full defense against the phages. And what's very interesting about this is that when you mutate either of the two branching guanosines or you introduce a mutation in the active site of the reverse transcriptase, you fully abolish this defense phenotype, indicating that you need a mature msDNA to be generated in order to have this defense by the retron. Additionally, as I've mentioned before, retrons are tripartite systems, and in this specific example, the accessory gene is an endonuclease, and similar to the mutations that I just mentioned, also if you mutate the active site of this endonuclease, there's no defense anymore that the retron can provide against in this phage. So this is very interesting and it was a super nice discovery, but we were wondering how retrons actually sense the infection, because at this point what we knew was that they do somehow provide a defense against infection, but we didn't know how they do it. And to study this in more detail, we performed what's called an escaper assay, where we basically perform plug assays on a bacterial lawn that does not express the defense system, and then again on a lawn of bacteria that express the retron. Now, usually what you can see on these plug assays is that you will find a few plugs that will form even though you perform this experiment on bacteria that express the retron. And this usually happens because these are phages that have mutations that somehow allow them to escape the defense by the defense system. So what we did is we performed this experiment and then isolated these potential escaper phages. And when you then perform plug assays again without the defense system, there should be no difference between the wild type phage and the mutant phages. But when you perform the plug assay on a bacterial lawn that does express the defense system, again, the wild type phage should be protected against, whereas these mutant phages, they overcome the defense system and have a successful infection. We then sequenced these phages here that were able to overcome the defense by the retron, and asked where are the mutations in the genomes of these phages, since the mutated genes might be the potential triggers that the retron system recognizes or senses in case of the wild type phage. And by doing this experiment, what we found is that a specific retron that we were interested in, which is called Retron ECO8, actually was overcome. So the defense by this retron was overcome by phages that had a mutation in the single-stranded DNA binding protein inside of their genomes. So we then wanted to know how this single-stranded DNA binding protein actually activates the defense by Retron ECO8. So here you can see a standard northern blot from a whole cell lyset of bacteria either expressing Retron ECO8 or not expressing the retron. And you can see that we can easily detect the msDNA on this northern blot in these cell lysets. And what we then did was we co-expressed the phage single-stranded DNA binding protein and performed pull-downs in order to ask whether we are able to pull down the msDNA of the retron together with the phage SSB. And this is exactly what we were able to do. So here you see the pull-down of the phage SSB, and we were able to pull down the msDNA together with the protein, which you can see on the Western blot down here. And when we performed the same experiment with the mutated phage SSB that we identified in our escaper screen, we were not able to pull down the msDNA of the retron anymore, indicating that the msDNA of Retron ECO8 directly binds to the phage SSB. And then to further study how the phage SSB triggers the defense by the retron, we co-expressed the Retron together with the phage SSB. And when you do not induce the expression of the SSB, there's no difference in bacterial growth, as can be seen here by this dilution series of bacteria between the bacteria expressing the SSB of the phage together with the Retron and the two controls. But when we then induced the expression of the phage SSB, what we saw is that this was extremely toxic to the bacteria that contain the Retron, whereas the bacteria not expressing the Retron or expressing the phage SSB that was mutated, they showed reduced or no killing, indicating that the phage SSB directly binds to the msDNA of the Retron, and this somehow then activates the nuclease, which is the accessory gene of the Retron, and the nuclease then degrades the host genome and thereby leads to abortive infection and thereby the killing of the cell and the protection of the surrounding bacteria from the infection. So with this, I want to come back to the slide that I showed in the beginning, showing this vast variety of different defense systems that we have discovered over the last few years. But what I haven't mentioned when I showed this slide is that except for one of these, all of them protect only against phages with DNA genomes. And that raises a very simple question, and this is, what happens to phages with RNA genomes? And this is one of the things that I'm interested in studying in my own lab. So what are RNA phages? So RNA phages are only represented by two different families of phages. One is the Würzburg, where we have around 20 different isolated species, and they are very small phages, some of the most simple viruses in general. They only have a small 4,000 nucleotides single-stranded RNA genome that encodes for 4 genes. And some of the very well-known examples of these phages are, for example, MS2 or also Q-beta. And then the second family of RNA phages that has been isolated are the cystoviruses, where we only have around 10 that have ever been isolated. And in comparison to these smaller single-stranded RNA phages, they are much more complex, and they have around a 14 kilobase double-stranded RNA genome that is segmented into 3 different segments. On these 3 segments, these viruses encode around 13 genes, and they are also the only phages that have a lipid envelope, making them structurally very similar to certain eukaryotic viruses, which is why they are often used as a surrogate to study packaging, for example. And just to give you a small comparison, when we just looked at tailed DNA phages, we know 74 different families of them and several thousand. I didn't actually find a proper number how many thousands of different of these tailed phages have been isolated today, but it's a huge variety. They have also very different genome sizes from 30 to more than 700 kb, and they also encode for a lot of different genes. And with this, I just want to say that basically we know very little about the biology of RNA phages in general, making them basically like a black box when it comes to the molecular biology of them. And this is exactly what we are studying in my new lab. So we are really interested in the molecular principles that underlie the interaction between RNA phages and the bacteria that they infect. So one of the things we're interested in studying is the defense against RNA phages. So as I've mentioned earlier, DNA phage infection can be sensed by a variety of systems, most importantly restriction modification and CRISPR-Cas systems that really are able to recognize the invading DNA of a DNA phage and then stop the infection by cleaving the genome. How this works for RNA phages and if it works like this for RNA phages is currently absolutely unknown. And we want to figure out how RNA phage infection can be sensed by bacteria and then stopped by the defense systems. Another thing that we are very interested is to identify how RNA phages actually regulate replication after they entered the host. And this is because when you look at DNA phages, since they have a DNA genome, they do a lot of transcriptional regulation in order to regulate their protein output. On the other hand, if we look at RNA phages, they also have transcription, obviously, but as I've shown you in my slide about RNA phages, they can either produce only 1 or 3 different transcripts depending on what family they belong to, meaning that they have to regulate their protein output somehow on the post-transcriptional level versus on the transcriptional level. And just to give a bit more detail to this, if we look at a DNA phage, they use a whole range of different promoters in order to regulate transcription over the time of infection. And so you can classify genes of DNA phages into early, middle, and late genes. And they use both the host RNA polymerase and also own polymerases in order to do this. RNA phages cannot do this, as I've mentioned, and they have to rely on post-transcriptional regulation. So one example is the phage MS2 repressor complex. That's a well-known example where you have an aptamer structure in front of the replicase gene of phage MS2 that can be bound by the code protein of the phage in order to stop the translation of the gene. And we are interested in studying how other RNA phages than MS2 or the standard model systems actually regulate the protein expression during infection. And with this, I'm at the end of my talk. I want to thank my lab. I want to thank also my postdoc supervisor, Rotem Sorek, and also Sharon Wolf, who helped me a lot during my postdoc with the electron microscopy. And obviously, I want to thank all of you for listening.
Thank you very much, Jens, for this really, really interesting talk. So we have some questions. The first question is there any parallel mechanism of this beautiful system in human?
Which system?
I guess this question came in the very beginning, the defense system.
Okay, yeah, so basically, as I've mentioned, this this Bacterial ISG15-like system, so this ubiquitin-like system that we discovered is working analogous to the human ISG15 system, which is part of the innate immune system. So basically, after viral infection, you have interference production, which stimulates the expression of ISG15 and also of the E1, E2, E3 proteins that are necessary to actually conjugate the ISG15 protein to targets. This system protects against a variety of viruses, although the exact mechanism of defense is not particularly well understood. But yes, so this is basically the analogous system in the human immune system.
Thanks. Second question is, would bacterial defense system also function in human gut and have some interactions with human?
Yeah, so I mean, this is only, we are only at the beginning of understanding the answer to this. But we definitely do know from metagenomic studies, for example, that also in the human gut, there's a huge variety of different defense systems that we think bacteria also use in the phage bacteria warfare that happens in the gut all the time. So if you look at the virome of the gut, you can see that our guts are full of phages, and obviously they are also full of bacteria. So there has to be some type of arms race that both of them actually are able to survive.
Thanks. And we have another question on phage genome. So is there any benefit for phages to have an RNA genome over having a DNA genome, or is it purely evolutionary remnant? Perhaps you may compare that to the viral RNAs and DNA and RNA viruses as well for humans.
Yeah, so this is actually a very interesting question, because if you look at the virome of eukaryotes, most of the viruses that infect eukaryotes actually have RNA genomes. For bacteria, this is somehow the exact opposite. So most of the phages that infect bacteria have DNA genomes. And evolutionarily, RNA viruses in general are considered to be older than DNA viruses. So one advantage that RNA viruses in general have is that they usually have a much higher mutation rate, which might also be a disadvantage depending, right? But this means that they have very small genomes that they can mutate very easily in order to escape, for example, immune defense. But on the other hand, obviously, this also leads to the production of a lot of mutant viruses that are not able to infect. And this is not the case for DNA phages, for example. They can also have much bigger genomes, so they can have more proteins, for example, that are actively able to overcome defenses. So I think there's no clear way of answering whether one or the other is better than the other, but they both have advantages and disadvantages.
Thank you, Jens. For time reasons, there is one more question by Aditya Kumapal, but perhaps you could type in your answer. Thank you,
Sure.
Jen. And now we leave the stage to Shanghai RNA Club. Thank you.
Hello, hello everybody. I'm Paul from Shanghai Tech University. I'm honored to introduce Dr. Ren Jie. Jie is the principal investigator at the Beijing Institute of Genomics, Chinese Academy of Sciences. In 2010, she received her PhD degree from State University of New York at Stony Brook, followed by postdoctoral research at Cold Spring Harbor Laboratory. In 2018, she joined the Beijing Institute of Genomics. Her research interests focus on the regulatory roles of RNA on chromatin to elucidate the mechanisms that prevent and resolve transcription replication collisions at various genetic contexts to protect genome and epigenome stability. To study this mechanism for better understanding of the molecular basis of cancer and aging, and to develop novel single molecule direct sequencing techniques. She has published a number of research articles in prestigious journals, including Cell, Nature, Molecular Cell, and Cell Stem Cell. So let's welcome her talk. The topic is Nascent RNA at the crossroad of transcription and replication. So, Jie, you can share your slides and we can start.
Okay, can you see my slide?
Yeah, no problem. You can start.
Okay, thank you, Chao-Po, for this very nice introduction, and thank you all for staying for this opportunity to introduce my work. So as Chao-Po mentioned, this talk is going to talk about nascent RNA at the crossroad of transcription and replication. So my lab study is nascent RNA. Why I think this population, this audience, I don't have to convince you guys that RNA does not just code for protein. But I want to mention also RNA, when they are just being produced, they can already carry out functions because they can be modified, and they can also form this so-called R-loop structure when they hybrid with the DNA template and displacing the other non-template DNA strand. So this structure will be at the heart of my talk. Well, this happens, transcription is not the only thing. This happens at the G2 phase. And during the S phase, when the cells are being replicated, if the PO2 are prolonged stalling for certain reasons, they might interfere with the progression of the replication fork, and that might cause genome instability. And we have other work to show that also affected the epigenetic memory, because this is the same stage when you want to pass along your epigenetic information to the next generation. So at the heart of our talk, we're going to talk about the nascent RNA forming R loops, which counterintuitively, because most cases, people view R loops are damaging as genome instability. Well, in this case, in certain cases, well, the R loop might have a different role as regulatory. But this is not new, actually. At the end of the genes, where you can see this is adapting from a very nice review from Proudfoot. At the very end of the genes, when PO2 has to stop, there are two stages. It has to first slow down and then release. And our loop highlighted here plays an important role in both stages. It not only pauses this fast-moving PO2, but also it has to be resolved so the PO2 can be released. Well, this role of our loop's dynamic role seems to be a shared scheme, actually. And well, now earlier study, well, looking at the double-strand break, where you can see the R loop forming at the beginning of the stage, actually is protecting the end not to be digested too much by the exonuclease. But when the damage has to be repaired by homologous repair to search for homology, the R loop has to be resolved to expose for homology. Well, similar cases has just been demonstrated at the reverse fork by two nice studies in molecular cell. Well, so this seems to be a common theme for R loop dynamics, but the question is what controls the pause and release of the R loops? So in this talk, I would like to share our findings with some initial study that stemming from this study that we established a connection between the M6A, which is a modification, the most prevalent modification on mRNA, and with the co-transcriptional regulation on this axis. Let's see. Well, the beginning of the story stems from the collaborative work with Yingui Yang and Qianwen Sun. So in this work, we found that M6A, this modification, is actually enriched more on the R loops rather than other regions of the chromatin-associated RNA. So we did this by fragmenting the genomics and pulling out the R loops by this S9.6 antibody, which is specific to the R loops, and then pass it along to mass spec and then ask for the fraction of M6A percentage. So we can see it's about 1.5-fold more enriched on the R loops. So now this seems like an intimate relationships. And moreover, you can see that the stronger, excuse me, the stronger M6A signal is associated with stronger R loops. So there is dosage effects on their strength and distance. And they are all concentrating at the usual place for M6A, at the 3' end of the stop codon region. Before we went into asking what the function is of M6A over there, I want you to pay attention to this blue curve. As you can see that the M6A modification is already installed to 50% of their level co-transcriptionally, because these are chromatin-associated RNA, meaning they're mostly nascent, just being made, compared to their mRNA counterparts, which are mature. So keep this in mind. We have half of the modification already there. The question is how, right? Before that, we move on to the function we ask here. Where are the M6A doing here? So for this purpose, we built a reporter system that has separated two luciferase reporters. And if this termination doesn't stop properly, it will read through into this blue luciferase, vanilla luciferase, and then you can use that as a signal for read-through products. What you can see is that removal of METTL3, which is the major writer protein of this M6A modification, where you see a great reduction of M6A, of course. What you can also see is an increase of the read-through products. And this increase of read-through products is depending on the M6A sequence. So if you mutate the A to and T, which is not able to be modified, then you will not be able to see this increase of read-through caused by knocking down of METTL3. So it seems like the M6A here is to preventing read-through in this case. So from this initial study, we established an intricate relationship between M6A and our loops. But there are still questions. First is what we have been asking is, how are M6A installed co-transcriptionally, at least half of the level, right? So how was that possible? How is that carried out by this most major writer, METTL3? The second question is, we know that M6A is concentrating at the termination region. What is its role there, and how exactly is carrying out this role? And moreover, how is that contributing to transcription replication coordination is what we have been wondering around. So R loops, often formed co-transcriptionally, seems to be a potential route. But there is a but, that we already know that METTL3, the major writer, does not like hybrids as their substrate. So in this case, means there has to be a protein X, a little helper, for this METTL3 complex to accomplish its role. And we also need to figure out how this modification is installed and regulation carried out in a step-by-step manner. So those three very brave graduate students at that time took on this project, and then let me walk you through. So what they have found is that when you use the components of the METTL3 complex as bait and pull down the interacting partners, what you found is a protein standing out called DDX21. I will keep the identity of this protein for later, but for now I would just say why it stands out, because it has the ability to recognize R loops. Well, of course, we confirm that this interaction is true with every component of the METTL3 complex. And moreover, when we use DNAs and RNAs to remove the nucleic acid, we see that the interaction is not disrupted, meaning that there are direct protein-protein interactions. So by using a bunch of truncation mutants, truncation proteins, we mapped that the interacting phases, the interface between METTL3 and DDX21 is formed by its N and C terminus. So if you only have one terminus, or if you lose both, the interaction is greatly compromised. And one more thing, since if you notice that there are nucleolar localizing signals on both termini, meaning that it actually localized to the nucleolus. So the question is, how does the protein localizing to nucleolus guide the M6A modification, which mostly happens in the mRNA? So we can, using the proximity labeling assays, we can see that the interaction between the DDX21 and the METTL3 is actually outside of the nucleolus. So whatever is outside of the nucleolus, the DDX21 is interacting with METTL3, and disrupting the nucleolar localization does not interrupt this interaction. But if you don't have the interacting interface, then you don't have the interaction. So they co-localize on the chromatin. Where, right? So it turns out they co-localize on the chromatin on R loops, because we know that DDX21 binds to R loops, and that is consistent with our previous observation that M6A are enriched on R loops. Well, moreover is, if you disrupt R loops by overexpressing RNase H, which is specifically digest away R loops, you greatly compromise the way of METTL3 can associate with chromatin. So it seems like R loop serves as an anchor for METTL3 to enter the chromatin. But DDX21, what is its role? For this purpose, we have used a global chromatin-associated RNA binding assay that use a METTL3 as the bait and pull down the chromatin-associated RNA with it and then blot it. If we have depleted DDX21, you can see the ability of METTL3 to pull down nascent RNA is greatly compromised. But that is not the other way around. And if you use ParClip to see sequence, which exactly the site of these DDX21 or METTL3 binding, you can see the same trend of nascent RNA binding compromised by depletion of DDX21, but not the other way around. And this recruitment to the nascent RNA is also greatly compromised by the expression of RNase H, disrupting the R loop to the similar level as you disrupt DDX1. So all that leads to a sequential recruitment. Is R loop serving as the footprint, the foothold for DDX1 to enter, and DDX1 recruits METTL3 so that it can see the nascent RNA. We reconstructed the entire system in vitro by assembling an R loop, and we can see the interaction between METTL3 and R loop are only present when you when DDX1 is also present. Of course, we confirm that this is a genuine R loop. It doesn't contain double-strand RNA. It contains some double-strand DNA because of the way it is formed, but rest assured that doesn't bind DDX1 or METTL3. So the final question, does it decide the M6A modification? Well, if it doesn't, it won't be sharing the story, right? Well, as expected, you disrupt either the R loops or the DDX1, you can see a great compromise in the overall level of M6A modification inside a cell. But remember that at the beginning of this study, we're starting with a dilemma that METTL3 installed M6A on R loops, but R loops is not its favorite substrate. Now it comes to the second identity of DDX1 rather than just a recruiting role, because it is also a helicase. The DDX21 is a helicase that can resolve the R loop and present the nascent RNA for the activity of METTL3. And if you disrupt the helicase activity by this mutant, point mutation, you will disrupt the, you will reduce the amount of M6A modification to the same level as depleting the DDX21 protein. So where are they? Before we go into that, we also in vitro reconstructed the system for METTL3 recruitment and methylation on the in vitro assembled R loops. What you can see is that only the wild-type DDX21 with the helicase activity intact are able to facilitate METTL3 for the modification on the R loops. Inside the cells, if you deplete DDX1, you can see a great level reduction of M6A, and that reduction is closely associated with the strength of the R loop, the binding strength of DDX1 and METTL3, and most importantly, and you can guess, that they are at the end of the genes. So again, the question, what is it doing there? Or more exactly, how this helping the termination there? Well, here using a model gene, actin B. Actin B is a model gene to study termination, and luckily it also has an M6A modification at its end. And more importantly, if you knock down DDX1, you can see the M6A modification is greatly reduced. That is because if you reduce RNase H, DDX1 recruitment to this site is greatly reduced. And that will reflect in a reduced recruitment of METTL3 at this site. And interestingly, if you look at the RNA product of this gene, what you can see is if you disrupt DDX1 or METTL3, you will see a longer transcript indicating a read-through product at the end of this gene. We also show that by using MNES-seq, which is a more precise sequencing method to show precisely where the PO2 is. And we can see a prolonged and increased PO2 presence after the gene ends. So there is a problem of releasing PO2. In the classical torpedo model for termination, XRN2 acts as the torpedo to digest and chase up the RNA and knock it off. So we look at our XRN2 and we see that if you disrupt any part of the axis at the beginning, you will end up with a great reduction of the recruitment of XRN2. And you can even see that our XRN2 and METTL3 are associated when you use the pull-down assay. And one interesting thing here, paying attention to the DDX21 helicase mutant. The helicase mutant are also able to disrupt the interaction between METTL3 and XRN2. Well, if there's only protein-protein interaction, the helicase activity is not needed. What needs the helicase activity is the M6A modification. And M6A modification is often read by a protein called YTHDC1. And we can see that YTHDC1 and XRN2 has direct protein-protein interaction, even in the presence of NRSA. So meaning that when the M6A is installed, it's read by YTHDC1 and recruiting XRN2. Once that interaction is formed, it is protein-protein interaction, is not bridged by the RNA. Okay, this is one gene. Actually, it turns out there are thousands of M6A plus genes. When you disrupt this axis, you will see an increased amount of PO2 downstream of the ending site. And this massive amount of, PO2 extending after the TES location, meaning that it might run into something else. For example, could it be running into replication and course damage? So we looked at the gamma H2AX signal, which signals for damage, and we see an increase in the absence of DDX21 or METTL3, and the cell survival are greatly reduced. And that increase of damage is depending on the helicase activity and METTL3, the methyltransferase activity. Again, highlighting that M6A modification is important for coordinating transcription and replication. But is that the case? If that is the case, we should be able to prevent damages by reducing collisions. And if you look at collisions, these are a two-party crime, right? So for one side, if we use CRISPR to inhibit PO2 downstream of TES, like using the CRISPR guide RNA1, you can see if we detect here that their level are reduced. If we used a negative control downstream, then detect here the PO2 is not affected. And when you inhibit PO2 from extending downstream, you can reduce the damage level. And the other side of the collision, if we use a very low dose of acetylcholine to slow down the fork, we can also alleviate the damage. And importantly, this damage, the signal, relies on the passage through S phase. So if we synchronize the cells into S phase or arrest them into G1, we can see that only those passaging through S phase with an active replication will endure with this damage. So we summarize this finding in this cartoon that the nascent RNA, like the yellow smoke coming out from the train, will pair with the track, the DNA template, forming R loops that allows DDX21 to recognize and bind. DDX21 recruits METTL3 and also using his helicase activity to present nascent RNA to allow the installment of M6A onto it. And the XRN2 sees this signal and know it is time to pull the trigger to stop the train. Otherwise, if you disrupt any part of the axis, you will have PO2 reading through that might cause DNA damage. So you can see at the heart of the crossroad between transcription and replication lies R loops, the structure, and the nascent RNA modification. And we step-by-step dissect how these R loops coordinate transcription and replication. Well, the question is, what also remains is how much it is conserved. What about a system doesn't have M6A? Where they will be functioning? And what about the other part, right? We focus a lot on transcription. What about the other part of fork? And what about the consequence of repair? And if it is not only transcription and replication, other events can also happen on the chromatin. This is a busy road. So what about the coordination of other events? Especially, we are interested to ask these questions in aging and other pathologies. So, well, one short answer is please stay tuned, that we are working on these part, and this paper is under revision. I would hope soon, fingers crossed, we'll be able to share some of our new stories on these. And I would like to thank all the collaborators, and especially the lab members who has carried out this work, and the people involved are highlighted here. And most importantly, thank you all for sticking to the end, and thank you for your attention.
Okay, thank you, Jie, for your systematic study on this. So the people have questions, you can put the question on the question and answer. So I have something to ask as a beginning. So do you see any phenotype of this? If a lot of RNA passing the termination sites. So what do you see any additional phenotype, like besides of the DNA damage response, and what happened to the cells?
A great question. A damage response is well, I would say an obvious one that we can look for, and there are also more consequences. So if you one thing you can, what we are guessing, and this is easy to think of, is that if you the PO2 runs more downstream, there is chance for it to use other polyadenylation sites, which are not usually available. And that may lead to a longer 3' UTR, and that will change the fate of the RNA afterwards. So that is some possible other kind of effects that may be observed. But I think that will need to be further investigated in specific pathology context for that. Yeah, but that's a great question.
Okay. I'm not so familiar with this area. So what's the collision, how the collision leads to the DNA damage response between the replication fork and the transcription machinery? I'm not so familiar with that. Can you?
Yeah, so if I can bring back my slides, I think that would be easier. Yeah, so that one actually helps. So here, if you have a PO2 stalling here, so there are two ways for collision. Showing here is actually a co-directional. A head-on will be even worse. But anyway, let's talk about this. So when we say a collision, it may not exactly be the polymerase and RNA polymerase where physically contact, because there will be topological stress ahead of it. And there's many other proteins that might be recruited. And also about the, so that signals for damage. That could be repaired and fork will be restarted. But even when the fork is restarted, you still have to remove the RNA polymerase. Otherwise, it will still be stuck, right? So that is one thing. But in the occasions, you can see that displacement of the single strand, which is often vulnerable, and that could be that could evolve into a damaged site, and the entire fork, if it is stalling for too long, it might collapse, and also other part of the fork might be reversed or evolve into a damage. That is also possible. So these kind of interference might not be a nice thing for the cells. So they want to be signaled and then want to resolve them. So what
I
we
see.
think is that there will be hub proteins that are being recruited and then coordinate the restart of the fork, removal of the PO2, and potentially repair the damage in a more faithful way.
Okay. So do you see there's any kind of very preferences of those genes that or just it happened ubiquitously to all the genes, or do you see some specific genes are more affected by the RNA methylation?
Yeah, that's also one question we ask ourselves. Is a very nice question. So in this case that we're sharing that this the genes are most susceptible are M6A modified. So it seems that these M6A modified genes might need more help. And you may think that even in situations or in organisms that doesn't have M6A, the genes doesn't terminate the same way. That's not a one-size-for-all story. Some genes may need more help, and there will be chromatin contacts that need to be recognized to help those PO2 to be released. Otherwise, it will interfere with replication or with other events, right? So I would think there would be a specialized, or not specialized, a specific group of genes that might be more susceptible, and they will still need, this is still hypothesis, but need to be tested if they can evolve into hotspots in cancer or other pathologies.
Okay. So thanks for your nice talk. And the next RNA collaborative seminar will be on March 26th, hosted by EMBO and the Italian Institute of Technology. So should I end this meeting or just give it back to the organizer? Okay, so thanks for everybody. We will end up this talk, and I'm looking forward to see you in the next RNA collaborative seminar.